So, hello everybody and welcome to today's Revive webinar by the Global Antibiotic Research and Development Partnership GARD-P. My name is Astrid Penzmo. I'm a Scientific Affairs Manager and I'm presenting today's session on antimicrobial drug discovery, SAR optimization and QSAR. Revive is GARD-P's education and outreach program, and it aims to capture essential R&D technical knowledge and share expertise with the global community through the Revive website. One main, but not only activities of ours are our webinars, all of which are recorded and remain available for free on our website. Also, today's webinar will be added early next week. A few other activities I would like to point out include our antimicrobial viewpoint series, which are short articles written by experts in the field, and our antimicrobial encyclopedia. This resource offers short definitions of key terms in the field, some of which are complemented by expert videos, and also most of them have relevant further reading links to external resources. As always, at the end of this webinar, we will have a Q&A session. You can send your questions at any time during the webinar by adding them in the questions box in your webinar control panel. And at the end of the webinar, we will do our best to respond to as many as possible. If at all possible, please also include the name of the speaker in your question. And I'm now happy to introduce today's panel. We will have presentations by Alastair Parkes from Evotec and James Duffy from Medicines for Malaria Venture MMV. And the session will be moderated by Charles Mowbray from DNDi. Charles is the Discovery Director at DNDi, that's Drugs for Neglected Diseases Initiative, where he's responsible for discovering and advancing new drug candidates into development. He joined DNDi in 2011 and has worked extensively on infectious diseases, including kinetoplastic diseases, with a particular focus on leishmaniasis and latterly also COVID-19. He is responsible for the drug discovery team and its activities, including screening, hit-to-lead, lead optimization, and preclinical and non-clinical activities in support of the DNDi portfolio. So welcome, Charles. I'm handing over to you to introduce our first speaker.
Thanks, Astrid, and hello, everybody. Great pleasure to be here today. At DNDi, we're always interested in learning more about approaches to drug discovery from infectious diseases. So it's a privilege to moderate this panel today. We've got two great speakers lined up for you. I think some thought-provoking, informative science to hear about. So we hope we'll have a good question and answer session at the end. Our first speaker today is Alastair Parkes. Alastair currently leads Medicinal Chemistry Department at Evotec, a global drug discovery and development company. Evotec's mission is to discover and develop highly effective therapies and make them globally available to the patients who need them. He joined the company in 2006, working first on central nervous system drug discovery programs and oncology projects before focusing on antibacterial research since 2014. Alastair's a regular presenter at medicinal chemistry and antibacterial drug discovery conferences, sharing results from Evotec's antibacterial research programs focused both on specific targets and also the development of new tools. He's been also a regular speaker on antibacterial drug discovery at Oxford University's Centre for Doctoral Training in the UK, and has also published a number of papers in this field. Before joining Evotec, Alastair was employed at the University of Manchester during his postdoc studies, and he holds an MChem and a DPhil from the University of Oxford. I'm now happy to welcome Alastair, and Alastair, over to you for your presentation.
Thanks very much, Charles, for that generous introduction. So today I'm going to talk about some aspects of SAR optimization in antibacterial drug discovery. And so I do have control of that. The various things I'm going to talk about. Firstly, I'll talk about the multiple challenges in antibacterial drug design. Then I'll focus on two of these specific areas, addressing access to drug targets in bacteria, and also designing for the site of infection, and I'll come on to what I mean by that in a few slides. And then I'll finish with a bit of a case study where we look at various different aspects which we've addressed in one program. So the first thing, what are the problems for antibacterial drug discovery? And I suppose the thing we need to think about is how do we design a compound which meets all the different criteria required? So we have properties to inhibit the biochemical target. We have properties to get that molecule into the cytoplasm of the bacteria, well, assuming our target is in the cytoplasm, and I'll be talking mostly about the idea of targets which are in the cytoplasm. And then lastly, the properties which you need to get to the extracellular compartment so that the compound can then accumulate in the bacteria. And of course, the properties to get to the extracellular compartment and also properties for accumulation are both multi-parameter problems in themselves. But ideally, we want to get to that central point where we have the properties which meet all these different criteria, and that's where the drugs exist. So in the past, there have been some attempts to identify or really define that space for antibacterial drug discovery as a whole. And back in 2008, a really important study showed that in general, antibacterial drugs tend to exist in quite different chemical space from other drugs for other disease areas, and that this, what you might describe as preferred antibacterial chemical space, is quite different from a typical screening collection. But later publications and certainly a lot of later studies have shown that while the antibacterial drugs might be different from the requirements for drugs for other disease areas in many cases, for different bacteria, the requirements can be very different. And so in a publication by AstraZeneca, they showed in terms of polarity that for different species, both gram-positive and gram-negative, but different species and different strains within those species, that the requirements are quite different. So the idea of a general antibacterial chemical space is probably not viable. And we really need to think about the different species we're targeting and enabling the compounds to get to the cytoplasm and to inhibit the targets in all of the species that we need to target for a particular indication. So I'll focus now on addressing access to the drug targets in bacteria. So this is really thinking about getting through the outer envelope of the bacterial cells. So now we're thinking about, in our schematic, the properties for accumulation in the cytoplasm. And I'll talk about the cytoplasm for the following reasons. If we think about the targets of most antibiotics, antibacterial drugs, most of these targets are in the cytoplasm. Perhaps the most successful drugs have targeted cell wall synthesis and repair, and there's probably a good reason for this, that probably slightly easier access to these targets. And in this gram-positive schematic, you can see that you've got a cell membrane, a cell wall. But if we move on to a gram-negative, you have the extra barrier of the outer membrane. And now you can see that all of the targets, the major targets, are within that outer membrane. So we've got to cross that, we've got to find a way through to enable accumulation at the site of the targets of the drugs we're trying to develop. So focusing on the outer envelope, and particularly focusing on gram-negative bacteria, we can look at this in a bit more detail. So at the bottom of the page, you've got the inside of the cell, and the top, the extracellular environment. And we start with lipopolysaccharide, which is a polar outer function, and this prevents lipophilic molecules from getting into the bacteria. On the inside, we have cytoplasmic membrane, which is a barrier to polar molecules. But what we're really considering is all of the different proteins, the transporters, that enable molecules to both get in and enable the bacteria to remove molecules that they don't want. So we have a number of different porins, we have different outer membrane transporters. In the inner membrane, there are transporters, there are efflux pumps, these are specific, some are general. And by creating strains of bacteria where these are overexpressed or knocked out, we can create really useful tools for understanding how molecules get in and out of bacteria, and start to develop guidelines and ways for us to design molecules to enable us to get into the bacteria, particularly into the cytoplasm, where many of the targets that we're trying to address reside. So keeping in mind at the top right-hand corner a schematic of the outer envelope, we can try to address the various different barriers. And so one of these is the efflux pumps. And for a long time, inhibiting these efflux pumps was thought to be a useful way. Well, it is a useful way to get molecules into gram-negative bacteria. And so two of the most commonly used are PABN and NMP, the structures shown here. They do effectively inhibit the efflux pumps and can restore activity against bacteria that express these pumps. However, these compounds, when dosed into humans, tend to have pretty poor PK and are often quite toxic. So rather than being useful in clinical treatment, these molecules tend to demonstrate more utility in determining efflux prevalence in clinical isolates, so more of a tool for understanding what's going on rather than a treatment. Another way to get molecules in is what's known as the Trojan horse strategy. So bacteria require iron for many cellular processes, and they secrete and reabsorb molecules which chelate iron. And this mechanism can be hijacked using this so-called Trojan horse strategy. And the first successful example of this is cefiderocor, where cephalosporin is conjugated to an iron chelating group. This enables the molecule to get into the bacteria, and this was the first antibiotic siderophore conjugate approved by the FDA, and this is for treatment of multi-drug resistant gram-negative bacteria. So this strategy certainly seems to be a promising one for getting molecules in. There are obviously other routes for entry into gram-negatives, and as part of a collaboration between Evotec and Harvard, we looked at conjugation with ciprofloxacin, and one thing we noticed was that these conjugates seemed to be gaining access where the molecules themselves were unable to penetrate the, well, we were looking at E. coli, but we think gram-negative bacteria slightly more generally. The way we looked at this was to take a fluorophore, which was shown not to accumulate in the bacteria. Conjugating the same fluorophore to ciprofloxacin enabled us to get in. What we haven't demonstrated with this is that we can get molecules into the cytoplasm, which then we demonstrate antibacterial activity through the target of those molecules. But I think this is an interesting observation and worth some more investigation. It certainly seems that ciprofloxacin is able to effectively drag other molecules, and sometimes quite lipophilic molecules, into the cytoplasm of bacteria. So that's something we'll look into in more detail. And then there are permeabilizers. So we have the polymyxins, and colistin is the one shown here, sometimes known as the antibiotic of last resort. These penetrate bacteria by effectively removing the barrier, permeabilizing the cell envelope. And this is interacting with both the outer and inner membranes. In terms of interacting with the outer membrane, colistin has many primary amines which are able to form cations, and these can stabilize metal ions, effectively removing them from the lipopolysaccharide, destabilizing the outer membrane and enabling penetration. Colistin also has a lot of lipophilic features, and these enable the molecules to act as detergents and disrupt the cytoplasmic membranes, but also other phospholipid bilayers. And this is where a lot of the toxicity from colistin arises. There have been attempts to generate newer polymyxins. Most of these are less active as antibiotics, but there are some with reduced toxicity which have been attempted at co-development for co-dosing with known antibiotics, because some of the less toxic polymyxin analogues are still able to permeabilize the bacteria. I put a link on here to a blog. Really, there's a lot of dislike for colistin among positions because of its toxicity, and so perhaps it shouldn't be being used anymore. And it's worth a read, the Perfect Storm blog at the bottom there about the dangers of colistin and why we should be looking at newer alternatives. While we have these tools, really understanding what's going on is really important to help us build further predictive models to understand how to get into bacteria. And so, as well as knockouts and overexpression of porins and efflux pumps, we can determine accumulation in bacteria in both whole cell assays, and this has been developed in quite a high throughput way, but also subcellular fractionation, so really understanding whether molecules get associated with the membrane, get into the periplasm, or get into the cytoplasm. And these assays are starting to come through in a more general way. So at Evotec, we've developed this on E. coli. We're also looking at Acinetobacter, but being able to understand where the molecules get to should help us to understand how to design for accumulation at the various sites of the targets. The subcellular fractionation assay is much lower throughput though, so building up this data will take a bit longer until we're able to make it more high throughput and faster. Probably one of the most high profile uses of accumulation data is the seminal paper by the Hergenrother group back in 2017. And I know there's a previous GARD-P webinar from this Paul Hergenrother, so I'll only summarize this in very brief detail. And if you want more information and a better summary of how this works, there is a whole webinar on this. But the key thing was that they measured accumulation in E. coli using the mass spec analysis of the cell material and the supernatant for a diverse set of molecules, and then used the data from that to try to predict what it is about these molecules that enables them to accumulate. And they found that positively charged compounds are more likely to accumulate than either neutral or negative compounds, irrespective of their polarity. Of the positively charged compounds that accumulate, primary amines were the majority. This may just be because primary amines represent the majority of positively charged compounds we make in drug discovery, but primary amines are certainly a useful tool. And then of the compounds containing the primary amines, rigid amphiphilic compounds and those with low globularity, and globularity sort of describes the kind of 3D nature of the molecule, they're the most likely to accumulate in E. coli. In a subsequent paper, the group shared a web-based application entryway. This was designed to be a simple tool to help predict accumulation. And they showed how addition of primary amines to a couple of molecules which already met the rigidity and globularity criteria resulted in broad spectrum agents. And so these papers are definitely worth a read. But the guidelines that more rigid molecules with some positive charge are useful for gram-negative drug discovery is well worth bearing in mind when designing new molecules. All of that research was done on E. coli, and so how about other gram-negatives? And there's a lot of data being published by the Zugorskaya group, and in this case they used wild-type hyperporinated, so reducing the outer membrane barrier, and efflux knockout strains, and those with combined hyperporination and efflux knockouts, to look at the diversity of the gram-negative permeability barriers. And so they used both MIC values of these different strains, but also accumulation assays. And they were assessing permeation, and efflux of a diverse panel of 47 antibiotics. The key takeaway for me from this is that the rates of accumulation of the various antibiotics vary significantly between the gram-negative species. So it's again more evidence that general rules for gram-negatives are not really likely to be successful. So they have different lipopolysaccharides, different porins, and different efflux pumps. So thinking about how to get into the various different gram-negatives, you really need to look at multiple species to have a chance of designing developing compounds which will enable you to cover the spectrum of bacteria that are needed for the indication. So if we move on to thinking about designing for the site of infection and going back to our schematic, bacterial infections will be in a particular compartment in the body, and we need to be able to get to that compartment. So we can understand the distribution by measuring tissue concentrations, but really designing for accumulation in specific tissues is very difficult. However, for urinary tract infections, we can focus on renal clearance to deliver drugs to the site of infection. And to do this, we can aim for small hydrophilic molecules with minimal plasma protein binding, and phosphomycin, drawn on the right there, a key drug for UTI, really meets all these criteria. So how can we apply this in a drug discovery project? Well, we were developing with a partner a series of compounds optimizing addressing a cytoplasmic target, and we were looking to design compounds for renal clearance. Most of our molecules are pretty lipophilic, quite high molecular weight, so we started to bring in groups giving us more polarity, trying to reduce the size as well. And what we found was changing these parameters resulted in weaker activity both at the target and in cells. But we were able to get more hydrophilic and smaller molecules that were still active. But the real question is, does this enable us to reach the urine in greater concentration? So some PK studies, compounds A and B, show that yes, this is definitely the case. The smaller, more hydrophilic molecule has a much higher proportion of the dose reaching the urine. It's also cleared more slowly, but the key thing is we were aiming to get the compound into the urine here. So our strategy seemed to be effective in terms of the PK. What we then need to understand is, how does this affect the activity clearing bacteria in mouse UTI model? So these compounds, they've got very different in vitro properties. We've got one which is more active, but a lower concentration in the urine. One significantly less active, but much more of it getting to the urine. And as you can see from the plot on the left, this describes a urinary tract infection model. So each dot represents a single mouse. And after 96 hours of treatment, we're looking at the concentration of bacteria, so the number of colony forming units per milliliter of urine existing after treatment. And what you find is both molecules have a significant reduction compared with the vehicle dosing, but these are not different from each other. And so having a higher concentration in the urine, we can have a much less active compound in terms of its target and still get the same result in terms of clearance from the urine. And then if we look at the other compartments relevant for urinary tract infection, the same is true. So these molecules, these molecules result in clearance which is not statistically different from each other in either the urine, bladder, or the kidney. And there's certainly a reduction in all of those three compartments. So the message here is that it's really the balance of properties that matters, and you may be able to come back a bit from potency to enable getting to the site of infection, particularly with urinary tract infections where you can design for that. But understanding the PK, measuring concentrations in the tissue may enable you to identify molecules which can give more efficacy in the in vivo models of infection. So I think I've just given these messages, but yeah, designing for renal clearance is possible. And so achieving high levels of renal clearance can result in enhanced efficacy. Okay, I'd like to finish with a quick case study thinking about a different target and how we've looked at various different parameters. in the terms of the design. So in this case, we're looking at LPXA inhibitors for Pseudomonas aeruginosa. And LPXA is one of the first enzymes in the creation of the outer membrane of bacteria, so gram-negative bacteria, I should say. So a very quick overview of what we've done here. We started with some hits from a Dell screen, and through some optimization, we're able to identify what I've referred to as compound 1 here. And the key thing about compound 1 was that it enabled us to get a crystal structure which showed that a particular group stabilized what had previously been a disordered loop, this flexible loop on the left-hand side. This information helped us to maintain the potency through different designs. And so thinking about our various properties for a successful drug, this is thinking about the properties to inhibit the biochemical target. From this, we had a potent molecule, but not much activity in bacteria. And what I describe as some sort of in-house knowledge, I think it's probably wider knowledge than just in-house at Evotec, but the addition of fluorines to aromatic rings seems to bring, or rather enable, activity against particularly Pseudomonas, but some other gram-negative bacteria. So the addition of fluorines gave us some molecules where our MICs were down in the single-digit microgram per mil level. But these molecules were still very unstable in vitro microsomes. So we've got a very short T-half. And so designing for stability and thinking about trying to get the molecules to the extracellular compartment, we're then thinking about MET-ID studies and identifying those points where we can make modifications to enable the compound to accumulate in greater concentration at the extracellular part of the bacteria. And so for this molecule, we were able to identify various metabolic liabilities. So replacing the sulfur, we've got hydroxylations at various points. Replacing the sulfur meant we needed to add an ethyl group to maintain activity, and this is coming back to the crystal structure. And so again, another MET-ID study, we needed to address hydroxylation on the ethyl group. And through a couple more modifications, we were able to get to a molecule with deuteration of the ethyl group to slow down metabolism. Also addition of polarity, another point in the molecule. By this point, we've got a molecule which is has a low MIC against a clinical strain and a much longer in vitro half-life, enabling us to look at some in vivo pharmacokinetics and predict the exposure in some infection models. So really, I just wanted to illustrate that the various considerations all need to be addressed as part of the SAR optimization. So I'll finish just with this this image. What we need to think of is antibacterial activities, but this is usually many different species, sometimes different strains. So we need to be able to think about the properties which will enable us to get into the point where these overlap. But on top of that, we've got all the different ADME criteria. And as we generate more data in all of this, we may start to be able to generate predictive models which help us to understand these. And so I think the message I want to leave you with is that certainly we've got some guidelines to exist, and the generation of data, the curation of data is enabling us to understand more about how to get to this sort of point where all the requirements overlap. So we are starting to be able to construct predictive models. The accuracy of those will improve as we get more data. And then once we have that multiple models for bacteria, the challenge is to combine with the multi-parameter optimization for all drug discovery. So with that, hopefully I haven't gone on too long, and I'd like to say thank you to the teams involved. We had very many people, so I won't go through them all, from Evotec, and we worked with teams from Harvard and our collaborators at Forge and Xbiotics. With money, I should say, from Wellcome and from Carvex at times. So thank you, and I'll look forward to some questions later on.
Thanks, Alistair. That was great. I'm sure there will be lots of questions. So thank you very much. Very interesting talk and interesting points for discussion later. Moving on, our next speaker is James Duffy. James Duffy is the Senior Director of Drug Discovery at Medicinal Malaria Venture, MMV, with more than 25 years experience working in drug discovery. He's got a special interest in malaria and other infectious diseases that disproportionately affect vulnerable populations in the Global South. Working at MMV, he's part of the efforts to reduce the burden of malaria in disease endemic countries by discovering, developing and delivering new effective and affordable antimalarial drugs. His current role is to provide scientific input and strategic leadership to multidisciplinary collaborations with academic, biotechnology and pharmaceutical partners engaged in malaria drug discovery. James obtained his PhD from the University of Sussex, and prior to working at MMV, he worked for Biofocus in Cambridge in the UK, where he was a co-inventor of the FDA-approved HDAC inhibitor, belinostat. James, welcome. Thank you for joining us today, and over to you for your presentation.
Well, thank you very much, Charlie. Okay, well, thanks a lot again to the organisers from GARP for the invitation to give me 20 minutes to talk about a topic that is dear to my heart, and this is really using computational tools to enrich our drug discovery pipeline, our portfolio of new antimalarial drugs, and how that we can use these tools and these methodologies to make the whole process more efficient. This is incredibly important whilst working in a resource-constrained environment. So that's fine. Okay, so this is to give you just a little bit of background where this project came from. So in malaria drug discovery, the cornerstone of our drug discovery efforts has been phenotypic asexual blood stage growth inhibition assays, and this has really been the real game changer in malaria drug discovery. It rather languished after, in the 80s, when really there were no one was actually interested in malaria as a drug, in terms of new drugs. But as resistance to the older antimalarials really took hold, and the case numbers and the mortality rates really rocketed, it became apparent that we needed to find new drugs with new mechanisms of action. Unfortunately, this coincided with the development of a much more affordable phenotypic whole parasite high throughput screening platforms, using a variety of readouts, a variety of parasite strains. But really, this enabled us to start testing millions of compounds. And I'm not saying, maybe it looks a little bit in my graphic, that we tested greater than 10 million compounds in 2008. That's not what I mean. What I mean, between 2008 and 2023, we've now tested probably over 10 million compounds. And this is with pharma, with academia, with research institutes, and even MMV, who've, through generous funding, have been able to acquire compound libraries and screen them. And from this, we've identified, depending on what your cutoffs are, somewhere around about 30,000 hit compounds, and this has progressed into more than 20 drug candidates from these phenotypic screening projects, and more than 20 novel mechanism of action. So as a means to an end, this has been a real game changer in terms of malaria drug discovery. But obviously, we want to try and leverage every possible bit of value out of these screening campaigns. Although they are affordable, they're still expensive operations to run. And is there more way that we can use the data? And this is really what led to the MAIP, or the malaria inhibition prediction platform. So why did we want to make this tool, and what does this tool do? I'll tell you. So essentially, the tool is what it says. It's a way of predicting the activity of a compound from its structure. It's a model for doing that, and there are a couple of reasons why we want to do this. So in terms of sort of infectious and neglected tropical disease drug discovery, we really obviously do need accessible and affordable and user-friendly computational tools. A lot of us aren't able to access maybe some of the state-of-the-art tools and models that are available to pharma companies. This may be because we're working in non-for-profit organisations or universities and things like that. So having these tools that can really sort of accelerate our drug discovery efforts and having them open access is really great. And the aim of this tool is to really maximise the impact of our compound testing by enriching the output, allowing us to select compounds with a greater likelihood of being active and getting more, bang for our buck out of our screening campaigns. We can think of many ways that we can use these sorts of tools, but typically we think of, we often approach pharmaceutical companies and say, well, would you let us screen a copy of your, a copy of some of your proprietary internal collection? And they may say, well, yeah, of course we can do this, but we're going to let you test it, but often it's blinded and often it's only a subset. So can we use this tool as a way to focus really in on the compounds with the best likelihood and probability of being active? Obviously, there are organisations such as MMV, and I'll give an example of this later, who are interested in designing and purchasing new antimalarial libraries, and especially when we have limited compound acquisition budget, we want to make sure that the compounds that we purchase are most, if they're relatively small libraries, the most likely to have antimalarial actives. And obviously you can think of using these tools. It's and I'll explain later, it's open access, and anyone who has a novel compound can simply plug the SMILES into the tool and it'll give a prediction of whether it's predicted to be active against malaria in an asexual blood stage assay or not, and MMV will test these compounds. And as a sort of a proof of concept, you can imagine this sort of tool being applicable not just to malaria, but against any other drug discovery project. I put NTD drug discovery projects, but theoretically any other drug discovery projects. But I guess that's why we did it. But what is it that we did? So, as I said, we took a 5 million compound subset of the compounds that were being screened in this assay. So these are generally from the screening projects. And as you can see, so we have a whole group of people who have some antimalarial data ourselves, and here I've given some examples of some pharmaceutical companies that have the data: AstraZeneca, Novartis, GSK, and there are others. And although many of the active compounds have been released, the actual full screening decks have not been released. For example, Novartis screened about 1 million compounds and they released the data for the actives. They didn't release all the inactives and, if you like, unblind their screening collection. But how do we actually how can we build a model that allows us to make use of this data? So the way that we did this was through a consensus QSAR model. So how does this work? Essentially, each of the partners, MMV, AstraZeneca, Novartis, GSK, builds a model for their compounds, and we define this by description. So it can be fingerprints of molecular structures, it could be calculated properties such as lipophilicity, it could be a variable, active, inactive. And so each of the compounds has a descriptor, and each of the partners builds a model from this. And each partner is completely responsible for designing that model and building it around their data. And then what we do is we essentially make a consensus model. So we take all of these models together. There are many ways that you can do this. You could take your compound and you could run it through all of these models, and you look at, say, the maximum score. So you generate the maximum score for each of the compounds you test, and then you rank them and you give them an overall score. You could do it as a mean score, or what we actually do is we do a meta model. And what this basically is, because this is less computationally intensive, it essentially defines scores for fragments of molecules, and we have I think it's about 1 million different fragments in the model. And this way we can run the model, and it basically gives you a score of the likelihood of your compound being active in the asexual blood stage of malaria. And this then you can use to decide which compounds to biotest or which compounds to synthesize. So this is all great, and this is all being published, and I mean the science behind it. I'm not the expert on this. Fortunately, the author of the paper, Nicholas, has actually done joined this course. I'm sure that he could answer any technical questions on this. But so we have this model, and this is great, and it works with test sets. So this is an example of how you use it. So here's a data set from PubChem. So obviously we have we have a set of compounds that we use to build the model, and then we have just a smaller test set. And the hit rate of the PubChem test set was about 0.4%. And what you can see here is basically the compounds in orange are the ones that were scored to be active, and the ones in blue are the ones that were scored to be inactive, or predicted to be inactive, or were inactive, and the ones in orange were active. And as you can see, as you get a higher model score, and this is using what the computational chemist uses, or the cheminformatician uses, this enrichment factor, which is the hit rate within a defined fraction divided by the total hit rate. And you can see that you get, as you get a higher model score, you get a higher enrichment factor, and you can see that you get a higher percentage of predicted actives. So this allows you to bias your compound selection towards compounds more likely to be active. And this has been published, and it's on the EBI website, and I put the link down there, and you can go along, and if you want to, you can put your compounds in there, and it'll generate a score. And based on our test sets, you know, we need prioritized scores above 40 or 50 for a good enrichment. So that's, if you like, great. So we built a model based on data. We've had some test sets, and we've proved that test sets kind of work. But I think people really, the value is when you actually have experimental validation of these models. So moving from a sort of a nice design and a hypothetical, if you like, example of this to actually using it practically. Does it work? Or is it just a quirk of the test sets, or how we built the models, and are we just finding out what we know? And, you know, all sorts of questions like that. You know, if you run the model, do you just pick other compounds that look like they're in the model? So we were lucky enough to be able to convince, you know, the management of MMV that we should actually use this model in anger and go out and purchase a set of compounds, and in parallel purchase a set of random compounds, selected using the same, if you like, drug discovery principles, but without the added MAIP prediction overlaid on top. So this is essentially the workflow that we use at MMV to design our compound libraries when we have the opportunity to buy new libraries. So again, working with the team at EBI, we use a reduced graphs and frameworks protocol. And this is basically a way of ensuring that the scaffolds that we purchase aren't present in compounds that we've already tested. As you see, we've tested, we tested about 10 million compounds, of which probably within the MMV database we have about 2 million compounds. So we're able to check that we certainly don't know that what the data is for them. We pick on diversity through clustering. This is primarily because again, really due to constraints of budget, we have to, if you like, spread our shots on goal as widely as possible. So we tend to pick compounds that are centroids or singletons from clusters rather than large clusters of compounds. As I say, we like to make sure they're novel, that no compound in the MMV is no compound in the MMV in-house database, or Kemble or Shaw Kemble. We have a list of fragments of existing antimalarial drugs, and we use this to ensure that the compounds aren't likely to be analogues of existing antimalarial compounds, which may or may not have drug resistance. And this is really important because obviously you can imagine with these sorts of QSAR type models, it's very good at predicting that something it knows about is going to be active. So if you were just to let the model run without the handbrake on, without any of these checks and balances, you'd probably find lots and lots of analogues of existing antimalarial drugs scoring very highly, because that's what the model does. But actually, if you apply a few filters, you still generate interesting new chemical matter that does have activity. So, and I'm going to show an example of this. we obviously look for compounds with balanced calculated physicochemical property profiles and potential for further development, no intrinsic structural alerts, if you like. And most importantly, we have, or at least as importantly, we predict their malaria activity through using MAIP. And this balance profile we encode using an MPO score. We're fortunate through generous support from the Gates Foundation to have licenses for StarDrop, so we use their MPO scoring system. It's not really I'm not really plugging this system as such, but it's a good way of us designing compounds with balanced profiles. So we avoid using strict cutoffs, and we try and select the properties to be complementary. So we don't put, you know, lipophilicity coded in two ways. We wouldn't have H-bond accepted H-bond donors and TPSA. So we try not to overscore for any property. We put most importance on avoiding structural alerts. We're actually, and this shows the MPO score for the library we designed to validate this model, and actually we're now more strict. We tend to only have compounds with an MPO of probably a greater than 8. That big sort of step you see where the values get orange is probably where they start picking up compounds with structural alerts. So probably the green compounds, which go from the ideal with the score of 1 to having compounds with, you know, lipophilicity outside of range, or, you know, more H-bond donors are accepted than we want. But the benefit of these kind of MPO tools is that they're soft cutoffs. You know, it doesn't have that kind of, oh, it's got a molecular weight of, you know, in our case 351, therefore it's bad. It just gets penalized for having. And this you can see is the overall balance of the libraries. So the compounds, you know, have how to say, physically chemical properties associated with good starting points for drug discovery projects. And as I said, the MPO's criteria is not met for all compounds, because we included, in this case, we included some high scoring MAIP compounds, because we really wanted to see how good the model was. So the next step in the process is really to plug this into our screening cascade, and we're really fortunate that we've been able to sort of build on a lot of the pioneering work from sort of 10-20 years ago, and have a really robust, what we believe is a really robust and comprehensive primary screening and hit triage cascade. We're lucky that we actually run the primary screen at one test center and then actually do the confirmation at another center. So we actually have independent laboratories confirming our data, and we have multiple assays. So we have drug resistant. So we start with a drug sensitive strain, NF54. We then confirm that in using a different. This is a luciferase expressing transgenic parasite. We then confirm that using another readout. This is an enzymatic assay using LDH fluorescence. The compounds that pass that criteria, in our case, it's 30% inhibition at 2 micromolar. We then generate concentration response curves, both in drug sensitive and drug resistant. Drug resistant, DD2 is a strain of malaria that's resistant to the current the older antimalarials. We also look at cell viability in a human HepG2 cell line. We're able to measure this assay at 12 hours and 72 hours, which gives us an idea of the speed of the action of the compounds. And obviously, as you can imagine, just from a clinical perspective, antimalarials that act quickly and bring down the parasitemia rapidly are much high value, if you like. After doing that, we then sort of assess, if you like, through the sort of chemist's eye of, do I like the compounds? Does it look like a project that we've already worked on? Does it look a bit, you know, that kind of thing? And then we simply repurchase the compounds and, I say, retest as a different test center. And what you can see on this graph is that the MAIP library, if you like, which is about 4,700 compounds, was tested in parallel with a random, set, as I say, designed exactly the same principle but without MAIP, of about 1,200 compounds. And you can see the enrichment that we're getting, even if you sort of normalize that across the across the various assays and as we progress through the cascade. The chemical QSAR library, as we call the library, does have a significantly higher hit rate than our libraries designed without MAIP. For example, we had a big library called the hit generation library that was about 140,000 compounds, and that had about 0.1% screening hit rate, which is about normal in our experience. It's somewhat difficult to compare with the original libraries that were screened 10 or 15 years ago, because I think that they hoovered up a lot of the low-hanging fruit, if you like. But on newer libraries, that's about what we get. And with this chemical QSAR library, we got about a 3% hit rate. And this led to 8 compounds which went all the way through the process and met our confirmed active criteria, which really is the start of sort of chemistry and drug discovery. These compounds all had MAIP scores of between sort of 50 and 60. 7 of the 8 compounds had MPO scores greater than 0.8, and at least in terms of chemical searching, they were for malaria. They were novel compounds. So you know, we really felt that this was a way of finding new chemical matter. And just I am a medicinal chemist, believe it or not, and so I do always insist on having at least one slide with some structures in it. And here are some slides. Here are some structures of the 8. Here are the structures of the 8 compounds. The actual MPO score, you can see there is actually one that we use once we've got biological data, so it takes into account the potency and the cytotoxicity, so the scores are slightly lower. But these are still reasonable starting points. Anything above 0.2 in our experience is pretty good as a starting point. You can see the potencies are all reasonable for antimalarial drug discovery projects. I think any of you that have worked on these sorts of projects know that hits around micromolar can translate into candidates with, with actual potential for single dose cure. You can see that there's no drop off in potency between the drug sensitive and drug resistant. 3D7 is the drug sensitive strain in this one. And importantly, the design principles have meant that we've got compounds which have got good solubilities and low clearances, which is associated with, which is a nice feature. Obviously, these can all be addressed, or can all be improved during optimization, but it's nice to start with compounds that have potential to deliver compounds or candidates with the super long half-lives that we need for antimalarials, especially within the prophylactic use space, where we're looking for compounds which have efficacious concentrations for about a month. So I hope in the last sort of 15 or so, 20 minutes, I've been able to give a flavour of a tool that we've developed that we think works well, is available to anyone interested in anti-malarial drug discovery, and hopefully acts as a sort of a template and inspiration for other projects that could potentially have collaborations with groups that don't want to share all their data or can't share all their data, it's too complicated to, but can actually build models based on their data, and how the idea of stronger together and putting these models into a single sort of meta model type architecture can actually deliver more. The sum of the total is more than the sum of the parts, is what I'm trying to say in a clumsy way. I'm sorry, there we go. Right. And as I alluded to during the presentation, we do have a commitment that if anyone out there has a compound that they think is that they have a sample of, they synthesize it, and it's novel, we're not going to test chloroquine for you, I'm afraid. We can share the data with you, but we're not going to test it for you. We would be happy to test the sample at no cost to the requester, and to share the results, and and hopefully kickstart some new malaria drug discovery projects wherever they are. Okay, so that just really leads me to thank the team, which, as you can see, the model was a team led by Nicholas Boss from EBI, and contained many pharma and academic partners in that. The experimental validation, thanks to the team at Pivot Park Screening Centre, who ran the assays, and TCGLS, who ran the follow-up work, and obviously my colleagues, and all the contributors, especially the Bill and Melinda Gates Foundation, who funded this project. as I say, this is, I think, the sort of philosophy of MNV, and I think most of us in the PDP arena believe that, you know, working together in partnership is really the only way that we're going to deliver on our ambitious goals. Obviously, thank you to our funders, and thank you. And it sort of looks a little bit like that without any snow in Geneva today, doesn't it, Charlie? we don't have any snow when it's 37 degrees outside.
Thanks, James. I believe it's now 39, so definitely no snow.
That's good to know. Good to know.
Thank you both. Terrific talks and spot on time. So that's great news because it means we've got time for some questions. So we're going to start the Q&A session in a moment. As a reminder for the audience, you can submit your questions as shown on the slide on the screen now. We'll do our best to get through as many of those as we can during the allocated time. So we've got a few already. If you've got questions in your mind, please jot them down and we'll be happy to try and tackle them. So whilst people are writing some more, there's some first few that have come in. So maybe I can pose the first one to Alastair. So question came in asking if you could comment on the degradation patterns for the ethyl and nitrile group to guide next step modification. So I think probably asking about the metabolism of those in your LPXA compounds.
Yes, so the ethyl group was both hydroxylated but also oxidized, so it became ethene. We didn't follow it further than that. I think there's a little bit more work to do on that for it to potentially become a candidate. So if it was a bit closer, we would probably follow up further and do some MetID studies based on in vivo samples. The nitrile, I don't know. We didn't have that. The replacement of the nitrile was more about trying to make the molecule a bit more polar. Generally, more lipophilic molecules tend to get chewed up more quickly, and so we're trying to sort of get away from that aspect. Yeah, so I think that's the main part of those two. The ethyl group was the liability where we really sort of identified the things that were going on in vitro MetID studies. So yeah, those are the things to look out for. And replacing with deuterium just uses the kinetic isotope effect. So the same processes will be happening, they'll just be a bit slower.
Super, thank you. And another one for you as well. One of our questioners is asking, what's your opinion of the future of natural product discovery for new classes of antibiotics using natural products versus in silico and structure-based drug design methods? Again, probably a question you hear many times in this field.
I think natural products have provided most of the scaffolds that have been successful antibiotics over the years, and so I think we would be very unwise to rule out the idea of identifying new starting points from natural products. I think, you know, we need to approach this from every angle we can, and so I certainly would encourage anyone who has novel structures, novel scaffolds from natural products to be looking at their antibacterial activity, particularly if they're easy to produce in large quantities and easy to synthesize and modify. You know, the downside to natural products is often the difficulty in developing SAR. So, you know, if you've made the molecules yourself in the first place, you know the routes, you tend to be able to optimize, modify, develop more data. For natural products, well, particularly with the beta-lactams, actually a huge amount of chemistry was developed to enable the optimization and the development of beta-lactams and beta-lactam analogs. So yeah, natural products, definitely I'm very enthusiastic about potential new natural products, but they have their downsides, as do compounds from screening libraries. Sure.
So no, thank you. Maybe one more question for you, and we'll take a few for James. Wondering, you highlighted the beneficial effects of fluorination in improving the activity of the LPXA inhibitors. I wonder if you've got any hunches why that is. Is that change in physical properties or something else?
My hunch would be that perhaps the bacteria haven't seen so many fluorinated things over the course of their evolution, but it is only a hunch. This is one of those things which medicinal chemists sometimes learn from talking to each other, where we put we put fluorines on these things, and it seemed to give us better accumulation. Physical properties, certainly there will be changes. There'll be changes to the electronics. I suspect the most likely thing is that they're just new to the bacteria, and so they haven't evolved their mechanisms for chewing up and getting rid of fluorinated molecules in the same way as they have for all the other molecules they've seen over billions of years.
Super, thank you. Don't go away. We've got some more for you. The queue is growing. but maybe we can pass the mic over to James and ask a few questions about your talk. And there's a question here saying, great talk. I fully agree. Have you looked at the 5 billion compound set from a different perspective to see if you could predict other parameters such as cytotoxicity or other things from this kind of meta modeling approach?
It's a great question, and we haven't done that with our data set yet. So obviously we do have other data that is generated. We don't, the primary screen of those is the asexual blood stage activity. The secondary screening set is much smaller. I mean, we do actually have sort of a curated set of compounds, all from the same assay. There's about 250,000. I think of those, just to give you an idea, the ones which actually have cytotoxicity is probably about 4,000 of those, and the ones with other properties like solubility, microsomal stability is about 120 of those. So it crunches. And the other thing is we're very fortunate. We've got partners, like AstraZeneca and so on, who will let us use their in-house models, their AI machine learning models and QSAR models to predict compounds. So it's a nice idea, but and I think the approach could be applicable, but we haven't done it with our data.
Okay, no, thanks, James. Another question. Can you comment on the success of not just finding new hits, but finding new mechanisms of action or pharmacophores that weren't represented in the 5 million training set? Could you? Yeah.
No, I can't. Right, and that's in a good situation, because one of our sort of key early discovery assays is to take these compounds and test them against a big panel of mutants raised against all sorts of investigational drugs. So you have a kind of a mutant associated with a mechanism, yeah, and that's really useful. It allows us to down prioritize the compounds with mechanisms we're not interested in or a lower priority, and maybe up prioritize, if that's the right word, I don't think it exists, the compounds with mechanisms we're interested in. But the other thing we can get, which is even better, is when we don't find anything and the mechanisms to us are novel, and that's really where we are at the moment. So the next step would be, once we actually put them into active drug discovery projects and have some confidence that they actually are projects with legs, we can then feed them back into groups where you can actually look at the target identification. So we haven't, so that was a long answer to say, no, we don't know yet, but I'm pretty hopeful that we will get some out of it.
Smash it. Well, great to hear that, and yeah, look forward to hearing more about that piece in the future. Yeah, I guess a question here, another question about natural products for you, James. So what's the prospect for using natural products for QSAR as antimalarial agents? So I guess, are they represented in the set? Can you start QSAR with that?
I mean, as those I suppose, even those of you who don't work on malaria, and certainly those of you do, will, you'll be well aware of the importance of natural products in the armory of antimalarial drugs. Artesunate is still a frontline antimalarial, and obviously, compounds such as quinine, with really the starting point of malaria drug discovery. You could argue much drug discovery, and they are represented in the data. Now, there won't be you know, you take a synthetic compound, and I don't know what's in the data set, but I'm sure the chloroquine analogues or something like that, there'll be hundreds and thousands of them, yeah. Whereas artemisinin analogues, there's likely only to be, you know, tens. So they will be in the model, but they won't be so well represented. You know, and I mean, and that is a reality of the model, is that if you know, if the compounds really are from novel classes of natural products, which do really do, as Alastair was saying, allow you to access parts of chemical space that you don't really get to any other way, you know, they may not be scored as highly, but hopefully they will still contain structural features that would be associated with antimalarial activity. So yes and no, if that's the right answer. So the worst answer, probably.
Thank you. No, it's I mean, in infectious diseases, natural products have clearly been important, but it's still quite a polarizing question depending on people's perspective. So no, thanks.
I can give you a view on whether we should or shouldn't start with them, but that's a different question.
Maybe come back to Alastair then. Someone's asking, could you explain the rationale for using Cipro to smuggle compounds into the cell? How does that work?
Well, the rationale for trying it was an observation from examining a lot of literature data. We were interested in hybrid molecules and examined a certain amount of the literature in this, and it just seemed that Cipro was doing something in these hybrid molecules. Often people were using hybrid molecules to try and address two different targets at the same time, to minimize resistance development. So theoretically, multi-targeting should make it harder for bacteria to evolve resistance. So quite a lot of people had done work on hybrid antibacterials, and many of these had used Cipro as one of the two ends of the bifunctional molecule. And most of the time, those molecules weren't active at both ends, so weren't active at both targets of the bifunctional molecule. So it was really born of an observation, and we wanted to see whether actually part of the observation was that molecules which you wouldn't expect to accumulate seem to be active at their target. So we started to investigate this using accumulation assays, and I don't have a good rationale for how Cipro does it. It may be the porin that Cipro usually goes through, and it's just got a really good uptake mechanism, and so gets pulled through, can pull surprisingly lipophilic molecules with it, according to our investigation. But no, I mean, I'd love to have the funding to do a more academic kind of study on this, but it's one of those things sometimes in drug discovery where you come across something, but you don't have the freedom to follow it up. Let's say, you know, our aim isn't necessarily to develop a really good understanding of that so much as to try to come up with a compound that could potentially become a drug. And so, what we found with the molecules, so we had a compound active only against gram positives. We got some activity against gram negatives with our hybrid molecules, and we showed that they were likely to be getting to the cytoplasm, but the gram negative activity was coming from ciprofloxacin activity. We were in gyrase inhibition. So yeah, I don't know if that actually answered the question. I just gave you a bit more of the story, but the key thing was that there's something going on with Cipro, and it seems to be able to pull molecules in. Whether that's useful is another question, and I'm not sure we've truly demonstrated that it's useful. It's just an observation that I like to share with people, because maybe someone will look into it a bit more and find out what's actually happening.
Yeah, no, interesting. And I just, as you're talking, I'm wondering if it's just penetration or if it's changing the residence time, if you like, the affinity for another target that's just makes it hang around longer in a dynamic process. I mean, it's clearly not an equilibrium with bacteria. There's things are being pumped out, but if it just makes it harder for the drug to leave, that might help as well, I guess.
Yeah.
Another tricky question for you, but I think antibacterial are tough. So could you give me some insights regarding designing and developing biofilm inhibitory compounds?
So that is a tough one. I've not worked on targeting biofilms specifically. I mean, breaking up biofilms, I don't know that we have really good tools for understanding what molecules need to do to get into biofilms. I know why the question is being asked, because biofilms are, a particularly difficult problem. I think many of the approaches that I've heard of have been different modalities. So thinking about things like antimicrobial peptides. But no, in terms of small molecule drug discovery, it's not something I've addressed, and I'm not aware of really good strategies for getting small molecules in. So I think I'd think about other modalities, but I guess testing and to understand whether your molecules have any use in that kind of problem is what needs to happen.
Okay, no, thank you. Perhaps we can bounce back to James again. Question: could you comment on, thank you, great talk, and the MAIP initiative is really exciting. Can you comment on the uptake? How is the community using it? Have you got good engagement and interest in actually applying the tool now you've developed it?
I'm going to give you another yes and no. We've had lots of views of the platform, people uploading compounds, so that's all pretty good. We use it actively. It's been used in the design of, I know it's been used in the design of other libraries. So that bit, yeah, it's good, and it's certainly stimulated people's thinking about these consensus models and how we can do that in a sort of interface between non-profits and commercial organizations and confidentiality and all that kind of stuff. What we haven't actually had is a lot of people coming along with saying, oh, I've got a compound and it scored really well and I'd be really interested in seeing it. That we haven't done, and perhaps that's probably our fault for not publicizing it as well as we have. And hopefully the work that I showed today is just about to be published, and hopefully it'll have a little bit more visibility. And from a selfish reason, that's why I quite like to do these webinars, just to make that plug for that. So again, it's sort of a yes and no answer.
No, great. And I guess the second part to that question is, do you think a similar strategy could be developed and used for other neglected diseases or other infectious diseases?
Well, I mean, again, simple answer is yes. I mean, I really don't see why not. I mean, with any of these kind of approaches, the more data that you have and the better curated the data is that you're using to build these models, the better the models are. So, so that would be, so if it's an infectious disease that's only had, a few hundred compounds thrown at it, it's going to be less effective than if there's millions of data points, I think. I mean, that's probably just stating the obvious. But I don't see why not. And all of the architecture, if you like, all the nuts and bolts behind it, is open access. So we developed it with the team from EDI with the purpose of making sure that there was nothing hidden in that sense. The only hidden bit is the compounds that GSK's corporate collection or Novartis' corporate collection, that you can't see, but you can see exactly how we built it.
Yeah,
I think there was a key point there about curation of data, because when you're expanding to other species of bacteria, and you'll often be able to scrape in quite a lot of data from the public domain, but really understanding whether the strains are comparable, or making sure that the database you're trying to build is homogeneous in a way that enables prediction, I think is really important. So that point about curation, I'd really like to echo that. I think it's vital when you're starting to build these models and build these databases to create the models.
No, I understand. Quality data and good curation's a challenge everywhere, especially with fragmented data sets and so on. So great. Maybe one last question each for you. Alastair, you've told us about the challenge of entry, especially into gram negatives, and the entry rules are a start, but as you said, they're still not general enough. You need to look at your particular pathogen and test against several if you really want to make sure you've got good entry. It sounds pretty challenging, and I guess there's the chemical space that synthetic and medicinal chemists operate in can be enormous. Your chemical space seems to me to be rather smaller. How do you feel? Is there still room to operate? Can you do enough? Is the space for new discoveries in innovation there, or are the walls closing in?
I think hopefully there is space for new discoveries. I didn't talk about a second paper from Helen Zugorskaya's group, where they've looked at Pseudomonas and tried to identify chemical space for Pseudomonas. And what they seem to pick out was that there's not one zone of chemical space where compounds will have potential to be active against Pseudomonas. There are various nodes, and we presumably haven't identified all of those yet because we've only tested a certain amount of chemical space. And so the encouraging thing is that there may be multiple different points you could get into to tackle different bacteria. Possibly the discouraging thing is that maybe it's harder to predict where those will be, because if these spaces are disparate, then perhaps we're going to have more trouble predicting them. But the understanding that there are other zones of physicochemical space and chemical space in general that we can get into and find activity, I think is encouraging. And of course, if we didn't believe that we could find new molecules, then we'd be in a lot of trouble. We've got to be optimistic about antibacterial drug discovery, I think.
I think that's something that unites us as medicinal chemists. You have to be an optimist because it's tough, but the creativity of medicinal chemists is endless, I think. So no, great to hear that. Thank you. Last question for James. I guess we've been hearing about, clearly many challenges in antibacterial drug discovery, but this one about entry being a really essential big challenge. Different from malaria. You've told us about the approaches to find the new hits and trying to do something different. When you do that, what do you think is the biggest challenge or the big challenge with those to turn them into drugs?
No, okay. I think malaria has some advantages over many areas of drug discovery in the sense that we have the assays are very robust. So we have very robust assays for parasite killing in the test tube, if you like. Those translate into pretty good animal models. We use humanized mice using the same parasites as we'd use in the test tubes, and we even moved to volunteer models where volunteers are inoculated with malaria. So we're able to, and we can test our investigational drugs and that. And the translation between those models is absolutely astonishing in the context of drug discovery. So in that sense, it's a cakewalk. Well, actually, it's not. So our big challenge is drug resistance. So propensity, parasites multiply so quickly. The risk of resistance is incredibly high, and so finding mechanisms that have low resistance risk, or having compounds that have polypharmacology that has low resistance risk is a massive challenge. Challenge number one. Challenge number two is, as I say, for treatment, we probably need to stay above the efficacious concentration for several days and kill quickly. For prophylaxis, which is the really unmet need, we're looking at staying above an efficacious concentration for over a month. So we're looking at half-lives of somewhere around about 200 hours. Yeah. So, and this is way outside of normal drug discovery space. So so we're kind of operating on the edge there. And just to put the icing on the cake, our target population, 80% of malaria mortality is infants, children under 5, yeah? And these are not the three of us talking here, and probably not anyone in the audience, I imagine, unless someone's brought their child along, and that we're not the same. And the actual designing medicines that actually are your target population are children and women of childbearing potential, mothers and pregnant women, is a completely different game in terms of the safety considerations. And then I guess, you know, just to finalize it all off, we're also looking at $1 a dose. So your cost of goods is going to have to be, very low. So we have the good and then we have the difficult. So I think you asked me for what the biggest challenge was, and I gave you 4. I don't know if that's you think about the question, Charlie. You know,
Fantastic, thank you. I mean, you've both talked about multi-parameter optimization because you've got multiple challenges, so well understood. Thank you. You know, it's clear that the job for medicinal chemists is far from done, and you know, you've I think given us hope, and your enthusiasm for using these new tools and tackling big problems like this is great. So wonderful, thank you very much. I'm going to close the Q&A session now. So many thanks to everybody who submitted questions. Sorry if we didn't quite get through them all, but I think we're about there on time. So hope we managed to cover most of them, and hand back to Astrid.
Great, thanks a lot, Charles. I think we did good with questions, and if not, we will follow up per email and maybe share a few more responses with the audience on our website in writing. So thank you for moderating the webinar so well and contributing, and also to battle the heat in our non-AC offices here. And then, of course, James and Alastair, thank you for your presentations and sharing all of your experience. It's really been a pleasure, I think, and we also received quite a few nice comments, which we will share in the follow-up. So, moving forward, I want to announce our upcoming webinars. We have a very busy webinar period coming up, which I'm excited about. So we have a webinar on the 5th of September about starting an antibacterial drug discovery screening program, followed by a webinar on the 20th of September, completely different topic on clinical trial platforms for new and neglected antimicrobials. And not even on this slide, we will have another completely different webinar on the 27th of September about antibiotic shortages, and more to come. So you can register for 2 of these webinars, and soon the 3rd one on our website, revive.carp.org/webinars. Oops, sorry. And to follow to stay up to date with our news and new webinars and other activities, you have different opportunities, the website, the newsletters. So everybody who registers for a webinar is signed up to our newsletter, and also on social media. And one thing which we will announce very soon is the upcoming ACC Antimicrobial Chemotherapy Conference 2024. We will soon open the registrations and share the draft program, which will be another very exciting step for us. All right, so this is all, and as I said, thank you to the speakers, the moderator, and also our audience for joining today, and I hope to see you again soon. Goodbye.
Thanks, everyone.
Bye, everyone. Bye.
Goodbye.